In this video lesson, we will see how observing an individual's phenotype is not sufficient for determining its genotype. Our objective for today is to be able to characterize the difference between an organism's genotype and its phenotype. And our vocabulary includes phenotype, genotype, and Punnett square. Things aren't always as they appear. Take skin coloration, for example. Humans and many other animals have a gene that contains the information for producing melanin, one of the chemicals responsible for giving our skin its color. This gene is one of many that influence skin color. Unfortunately, there is also a defective, non-functioning version of the melanin gene that is passed along through some families. An individual who inherits 2 copies of the defective version of the gene can't produce pigment and has a condition known as albinism, a disorder characterized by little or no pigment in the eyes, hair, and skin. But it's impossible to tell whether a normally pigmented individual carries one of these defective alleles just by looking at his or her appearance. We would need to get a genetic analysis done to discover this information. The inability to deduce an individual's genetic makeup through simple observation is a general problem in genetics. Physical appearances don't necessarily reflect the underlying genes. A normally pigmented individual may carry 2 copies of the pigment-producing allele, or they might just have 1. In either case, the individual will look the same. The outward appearance of an individual is called its phenotype. A phenotype includes features visible to the naked eye, such as flashy coloration, height, or the presence of antlers. A phenotype also includes less easily visible characteristics, such as the chemicals an individual produces to clot blood or digest lactose. An individual's phenotype even includes the behaviors it exhibits. Underlying the phenotype is the genotype. This is an organism's genetic composition. We usually speak of an individual's genotype in reference to a particular trait. For example, an individual's genotype might be described as homozygous for the recessive allele for albinism, meaning that the person has 2 recessive alleles of the gene. Another individual's genotype for the melanin gene might be described as heterozygous, which means it has a recessive allele and a dominant allele. Occasionally, the word genotype is also used as a way of referring to all of the genes an individual carries. When an organism exhibits a recessive trait, such as albinism, we know with certainty that its genotype is for the melanin gene. When it shows the dominant trait, on the other hand, it's impossible to distinguish whether the organism carries 2 copies of the dominant allele, meaning it would be homozygous dominant, or if it carries 1 copy of the dominant allele and 1 of the recessive. Of course, that means it would be heterozygous. Because it's not possible to discern the genotypes of 2 individuals with the same phenotype just by looking at them, much of genetic analysis must make use of clever experiments and careful record keeping. To analyze and predict the outcome of crosses, first we have to assign symbols to represent the different variants of a gene. And generally, we use an uppercase letter for the dominant version of the allele and a lowercase letter for the recessive version of the allele. In the case of pigmentation or albinism, we use the letter M because the trait is caused by the gene carrying instructions for the production of the pigment melanin, so M for melanin. We represent the genotype of the albino giraffe as m because that individual has to carry 2 copies of that recessive allele, m. A giraffe that is pigmented, though, must have the genotype of big M or big M. Now, if we don't know which of the 2 possible genotypes the pigmented individual has, we can simply write big M underscore, where underscore is a placeholder for the unknown second allele. We can trace the possible outcomes of a cross between 2 individuals using a handy tool called the Punnett square. Now, I emphasize the word possible because these are probabilities. The Punnett square is all about probabilities. In the image you see here on the screen, we illustrate the cross between a true breeding pigmented individual, big M, and an albino, m. Along the top of the square, we list individually the two alleles that one of the parents produces. And along the left side of the square, we list the two alleles that the other parent produces. It doesn't matter which parent's on top or which parent's on the side. And then we split up an individual's two alleles this way so that although the individual carries two alleles, they're only going to pass on one of the alleles in either the sperm or the egg that it produces. So the two gametes that come together at fertilization produce the genotype of the offspring. In the 4 cells of the Punnett square, we enter the genotypes of all the possible offspring resulting from our cross. Each cell contains one allele given at the head of the column and one allele at the left of the row. In cross 1, illustrated in the image you see on the screen, every possible offspring would be heterozygous and would be normally pigmented because it receives a dominant allele from the pigmented parent and a recessive allele from the albino parent. In the bottom half of the figure in this illustration, cross 2, we trace the cross between two heterozygous individuals. Now, note that each parent produces two kinds of gametes, one with the dominant allele and one with the recessive allele. This cross has 3 possible outcomes. 1/4 of the time, the offspring will be homozygous dominant, or big M, big M. And another quarter of the time, the offspring will be homozygous recessive, or little m, little m. And the remaining half of the time, the offspring will be heterozygous, meaning big M, little m. Now, phenotypically, 3/4 of the offspring will be normally pigmented, big M, or big M, little m, and 1/4 will be albino, or little m, little m. Now, keep in mind, something that's very important here is this does not mean that if these 2 giraffes give birth to 4 different offspring, it does not mean that at least one of them will be albino and the other 3 won't be. What it means is that there is a 25% chance each time they give birth that the offspring will be albino. So this is all about probability and chance.